Good evening. Welcome to the new session of Pre-Med Pro. And today we are going to start with the class 12 botany, the molecular basis of inheritance. It is a very demanding chapter from you all of you guys, and I have received many comments regarding to start this chapter. So today we will start this interesting chapter called molecular basis of inheritance. So give us the topic, molecular basis of inheritance. Molecular basis of inheritance. This chapter generally deals with basis of inheritance. This chapter generally deals with the DNA structures, the functions of the DNA and RNAs, basic functions of DNA and RNAs, and with some functions of the proteins as well as the DNA fingerprintings. Okay, so we all know that DNA is a genetic material. So let us write DNA a genetic material. DNA, it is a genetic material. It is a genetic material. Okay, now what happens? DNA is a genetic material and it was been discovered by Frederick Griffith. So Griffith in 1928 said that DNA is a genetic material, but he cannot prove that DNA is the matter that passes from one generation to another generation. That DNA is the genetic material has been proved by him, but he cannot prove that DNA carries the information from one generation to another generation. Okay, so what did the Griffith took Streptococcus pneumoniae, a bacteria, Streptococcus pneumoniae, a bacteria. Okay, so after taking the bacteria, it is a rough strain, that is R2 strain. R2 strain and S strain. R2 is virulent and S is non-virulent. Okay, R2 and S3 strain has been taken by him. These are the two forms. Okay, two forms. Okay, now what did he do? Let it be the R1 strain and this is be the R2. Okay, now this is the R2, this is the S3. Okay, this contains a capsule. Capsule is the coating of the bacteria. Capsule made up of polysaccharide. It is a sugar. Polysaccharide is a sugar. Now what happens? The colony cell is rough in R2. It's rough, that is not shining. It is a not shining structure. And whereas the S3 is smooth appearance. Okay, this is shining. Now what happens? It is made up of capsule. This capsule of R2 is absent. In R1, S3, it is present. In S3, capsule is present and is made up of polysaccharide and is a kind of sugar. Okay. Now in pneumonia, it's a non-pathogenic, non-virulent, no. Well, it's pathogenic. In S3, it's pathogenic and virulent. Yes. Okay. Now what happens? In case of mice, that is the plus R2 strain. R2 strain is good. No pneumonia. Yes, no pneumonia. But no death of mice, no death. While in mice with S3 strain, S3 is virulent and it causes pneumonia. The mice dies. Again, in the third chain, here is the matter. The mice is infected with S3, heat killed. So here we see no pneumonia. S3 active causes pneumonia, but when the S3 is heat killed, it doesn't causes pneumonia. So no death of mice. Okay. Now again the mice is infected with R2. R2 is a good bacteria, but R2 and with S3 heat killed. So R2 is a good bacteria that doesn't kill the mice, and the S3 when heat killed, it also doesn't kill the mice. But in case we mix the R2 and the S3 heat killed, we see in the observation that the mice got infected with pneumonia and dies. Death. Okay. Now what happens? He was expecting no death. But mice died. He was expecting no death, but the mice died. Okay. Now what Griffith said. Something is transforming it. Something is the transforming. This is very important. Transforming principle / genetic material. Transforming principle / genetic material. Transfer from heat killed. Transferred from heat killed S3. S3 was heat killed, as we have seen in the first that being S3 the heat killed, it doesn't kill the bacteria, but when it is mixed with the R2 variant, it kills the bacteria. So there is something is transforming from the heat killed S3, because the S3 variant is generally poisonous and virulent. So heat killed is treated as non-virulent, but here something is transforming from the heat killed S3 variant to R2 bacteria. R2 bacteria. So R2 developed capsule. R2 also developed the capsule and changed into living S3. Which killed the mice, but he cannot prove it. What has been transformed, he cannot prove it. Okay, he could not prove it. Take it as a headline. Okay, what Griffith has said may come in the examination. See, these are the parts where the students don't give any interest because they think, oh, these are easy. So the question setter makes question from this part, because this is an exam not to give you admission. This is an exam to eliminate you from the competition. Okay. So now the biochemical characterization of the transforming principle. Give the heading, the biochemical characterization of transforming principle. Biochemical characterization of transforming principle. What will happen here? It was discovered by Avery, MacLeod, and McKenzie and McCarthy. Avery, MacLeod, and McCarthy. So what happened? Filtrate of S3 heat killed. Filtrate of S3 heat killed bacteria here. See what happens. Here the DNA, protein is not there. Protein, now RNA is there. So what happens? It is there. It changes to change. Now what happened? It digests the protein. Digestion of protein is made by protease. Okay. Protease is an enzyme which digests the protein. Protease, and it's an enzyme. I write it at E. Now the R2 variant, it's a living variant. R2 is the living variant. The R2 variant now changes the DNA protein. Here RNA is not there. And it is being digested by, the RNA has been digested by, who has digested the RNA? RNA has been digested by RNase. RNase is an enzyme which digested the RNA. Okay. Now what happened? Now the R2 is living here also. It is in living state. Now what happened? This DNA is digested. Now the DNA has to be being digested. No DNA, and it has been digested. Protein is there. RNA is also there. The DNA is digested by the enzyme DNase. Then enzyme digests the DNA. Now it is C and it is B. Now what happens? A, B, C. Now A, what the change we see in A? R2 living, the R2 living state transformed into, so changed into S3 living. Protein is not the genetic material. Thus, we can say the protein is not the genetic material. Okay. Protein is not the genetic material because on the A, we do not have protein. So, protein is not a genetic material. Okay. Now in B, R2 change into S3. It is also living. Now we say RNA is not also, RNA is not a genetic material. A genetic material. And in C, what C? In C, no S3 found. Only R2 found. R2 not change into S3. No transformation. No transformation. So we can see in case number C, the DNA is the genetic material. Okay. Now what happens? The confirmatory test has been done by Hershey and Chase. So who has confirmed it? Hershey and Chase. Hershey and Chase are the both scientists in 1952. What they have done is unequivocal proof that DNA is the genetic material. This came in most probably in Neet 2020 or 2021. Okay, virus T2 virus infection bacteria, that is bacteriophage. It contains dsDNA. And in third, the E. coli. So the conclusion we got P as DNA and S as protein coat, where let it be the T2 fudge, let it be the T2 fudge, DNA, it is the phosphorus, AMP, GMP, CMP, TMP, and this is the head and consists of the protein coat. This protein coat consists of the amino acid. Amino acid is made up of methane, methionine and cysteine. Okay, these two amino acids are present here, and so sulfur containing AA are methionine and cysteine. Okay. Now some T2 virus grow in a medium. Some T2 virus grow in a medium containing radioactive S35 enter into protein coat. So what happens? So what happens? Experiment was done. This contains the radioactive sulfur. Okay, this contains the radioactive sulfur. Here it contains the radioactive sulfur S35. Okay, now some in experiment we generally does, one is infection, the twice is the blending, that is mixture, and the third one is the centrifugation. Okay, so the first one is infection, the second one is the blending, and the third one is the centrifugation. In some T2 virus, grow in a medium. The T2 virus grow in a medium containing P35, phosphorus 35, oh sorry 32, phosphorus 32. Okay, P32. The T2 virus growing on a medium of P32, and it contains the P32 radioactive bacteria contains P32 radioactivity. Now what happens? So what happens after that? After that we see the bacteria containing sulfur S35. One is infection by E. coli and here it is a test tube. Let it be a test tube. Okay, and this one is contains here, this one contains with phosphorus P32. Both are radio blood virus that infect E. coli bacteria. Okay, now we have what happens into blending, and the third one is centrifugation. Now what happens here? Centrifugation. We remove contact between the virus and the bacteria. We remove the contact between the virus and bacteria. What we found after removing the contact between the bacteria and the virus, we found that the protein coat here, present in sulfur, the supernatant radioactive, that is radioactive S35, protein coat not enter into bacteria because no radioactivity in pellet part. पिलेट पार्ट में कोई रेडियो एक्टिविटी नहीं है. So पिलेट पार्ट. So protein is not of the genetic material. Protein is not a genetic material. In the other case, here DNA of virus enter the bacteria. Enter the bacteria. Okay, so what we see, radioactive P32 present in the pellet portion. From this experiment, unequivocally it has been clear that the genetic material is the DNA, because DNA contains phosphorus and it's not a protein, because protein doesn't contain phosphorus, it contains a sulfur. So if we do not get sulfur part in the transformed element, that is, we can say the phosphorus part, the presence of the phosphorus part, the radioactive phosphorus part in the resultant unequivocally says that the DNA is the genetic material while the protein is not the genetic material. Okay, so till then please go to this part that I have described, because this chapter is a bit hard, a bit conceptual chapter, and question comes from this chapter in every year. Okay, till then if you find any difficulties, write in the comment box, and today and tomorrow I will start from the next part of this chapter. Okay, so till then, good night, and please subscribe this channel, please like it and share it with your friends. Thank you.